Hi everyone, this is Heather Benson and I am recording a video to explain how to do the exam 3 written problems for Bio 375. And so I'm going to share my screen so that we can go through the problems and you can see my work. Okay, great. So just zoom in here so you can see. So the first problem asks about different diseases from class, and it wants to know different things about them. So part A asks which of the above terms is associated with Patau syndrome? And for that, the answer is aneuploidy, trisomy, and non-disjunction. And that is because Patau syndrome is a trisomy on chromosome 13. And so that would cause non-disjunction and the aneuploidy and then the trisomy. Part B, which of the above terms is associated with the conventional form of Down syndrome? And that is chromosome 21 has a trisomy. So same thing as part A, just on a different chromosome. So the answers are the same: aneuploidy, trisomy, and non-disjunction. For part C, which of the above terms is associated with Turner syndrome? And that is aneuploidy, monosomy, and non-disjunction. And that is because Turner syndrome is where a female has just one X chromosome instead of two. And so that corresponds to monosomy, which is abnormal. So aneuploidy caused by non-disjunction. Part D, the answer, or the question says, which of the above terms is associated with Cry-du-Chat syndrome? And that would be deletion. And that's because Cry-du-Chat syndrome is where part of the chromosome number 5, its P arm is, part of it is deleted. And so that's a deletion. So on the next page here is part E. Just get this so you can see. So up here, which of the above terms is associated with type A hemophilia? And the answer to that is inversion. And that's because type A hemophilia is when the factor VIII gene has an inversion on it. Okay, so moving on to number 2. Number 2 gives us a sequence for E. coli, which is a prokaryotic organism. And the first question asks down here, after it gives us some information, it gives us 2 RNA primers, and it tells us this newly synthesized DNA strand will be synthesized as Okazaki fragments, which hints to us that this strand here is the lagging strand. And so indicate the polarity of the primers on the image shown above. So the answer to that is on the left side we have 5' and on the right side we have 3'. And that is because the corresponding lagging strand, the way that it matches the lagging strand, shows us that it has to be 5' on the left and 3' on the right. So for part B, what is the DNA sequence that will be synthesized between the primers? And be sure to indicate the polarity. So I've indicated that here, 5' CGA, GCTA, A, CT, CCA, GG, CA, C, GC, 3'. And the corresponding bases to the primers A and primer B match up with the sequences in the given DNA lagging strand right here. So you can see how I wrote that below on each of these and then mapped them up to find them to make sure that that was correct in the lagging strand, which it was. So for part C, which direction, right to left or left to right, is the replication fork moving as this daughter strand is being synthesized? And that would be right to left. The reason for that is because we know this because the way DNA is replicated, the leading and lagging strands are facing in opposite directions. And so DNA is synthesized 3' to 5'. And so because of that, if the lagging strand that they give us up here is going left to right, then we know that the leading strand, which has to be going in the facing the opposite direction, would be the direction that the replication fork is going. So that means we're moving right to left. For part D, which of these 2' was synthesized first? And that kind of goes along with what we said in part C, but because this is the lagging strand that we're talking about, primer B would have to be synthesized first. And that's because we're talking about Okazaki fragments, and so primer B is synthesized going this way, and then it waits for the helicase to continue splitting the replication fork further so that it can continue synthesizing, and next thing it synthesizes primer A second. Okay, so let's go over here to part number 3, question number 3. It says, suppose that an individual is heterozygous for the following change in chromosome structure. And then it gives us a normal chromosome and an inversion chromosome with the centromeres here. Suppose that these 2 chromosomes replicate prior to meiosis, producing 4 total sister chromatids. The 2 replicated chromosomes then attempt to synapse during prophase 1. What is the allele and centromere composition of the 4 products of meiosis if there is a crossover between T and U? So to figure this out, what I did was drew out the 4 total sister chromatids that were formed after replication. And so first what happens is that both chromosomes are replicated, so that forms the 4 here. And what's happening here is all the chromosomes tried to line up. And when they lined up, this top one went like this, and so the bottom one has to form a loop to try and match up to the top pair. The bottom pair has to try and match up to the top pair. And so in order for it to fit in, it has to form that loop, and the loop causes a crossover. So it tells us that the crossover is between T and U. So that's what I've shown here. And so this drawing helps us to figure out the 4 products. And because the centromere isn't inside where the inversion is happening, we know that it's paracentric. So right off, we can tell that there will be 1 dicentric and 1 acentric chromosome. So let me see if I can zoom out a little bit here so we can see what's happening. Okay, so chromosome number 1 will end up being the same normal chromosome that was listed above. And we can follow along on this top chromosome, and we have capital LMN centromere, capital OPQRSTUVWX. And that's the normal chromosome that's listed above. Number 2 is the inversion chromosome that's listed above, and the same thing. We can follow along here and go LMN, lowercase, centromere, lowercase, OPQRWVUTSX. Now for number 3, this is the one that will be the dicentric chromosome. And so what we do is we start here where I've indicated number 3 on this strand. And we go, I like to start from this side, because that just makes more sense to me. And so what you do is you do LMN OPQRS TUVW RQPO centromere NML, and I forgot to mention the centromere here, but that's listed down here with the dots. And that's a dicentric chromosome. And then number 4, that's on this strand here. And again, I like to start from where the capital letters are. And so what we have is we have how this corresponds here. And Anywho, so we have X, W, V, U, T, S, X. And so that's the acentric chromosome. And so it's important to note that the products 1 and 2 here will be healthy. Those are the ones that we started with. But products 3 and 4, the dicentric and acentric chromosomes, will be unhealthy because of the way that things worked out with the crossing over. Okay, so moving on to number 4. Suppose that the DNA molecule shown below is a portion of an E. coli gene. This E. coli gene is transcribed from right to left. The +1 site is shown in bold. So we have this here, and I just indicated the +1 site here. Part A says which strand, upper or lower, is the template DNA strand, and which strand, upper or lower, is the coding DNA strand. So the template DNA strand is the upper strand, and the coding DNA strand is the lower strand. And we know this because of the location of the +1 site. DNA always looks at the information of the strand 3' to 5', but the way that it replicates is the opposite. It goes 5' to 3'. So what we do is we start at the +1 site on the template strand here, and so we know that that means that the template strand has to be upper and the coding strand has to be lower. So for part B, it asks, what is the sequence of the mRNA molecule produced from this DNA molecule? And so we have 5' AGC, CUG, AAG, UUA, AUA, UG, 3'. And we know this because of we start at this +1 site up here on the template, and the template goes to the left because of the 3' to 5' directionality. So for part C, in which direction, left or right, along the coding strand relative to the +1 site would the -10 and -35 sequences be located? And they would be located to the right. And this is because TF2D and some other general transcription factors attach at the -10 region, which is where the TATA box is. And we know that that has to be before the +1 site where replication begins. And so because the directionality is 3' to 5', we know that they have to be located on the right side. Part D, in which direction, left or right, along the coding strand relative to the +1 site would the terminator be located? And it would be located on the left side of the +1 site, and that's sort of for the same reasons as part C. But the terminator tells the DNA when to stop replicating. So it has to be after where it starts. And so where it starts at the +1 site. And so if it's after and it's going in the same direction that we were saying in part C, then it has to be located on the left because it's on the opposite side of the +1 site as the TATA box at the -10 and then also the negative. Got it. So let me turn off the screen share here. Thanks for watching.